Imagine trying to understand how a major city works, right? When you know everyone's profession, but you have, like, no idea where the hospitals, the police stations, or the coffee shops are.
Right. You essentially just have the phone book.
Exactly. You have the phone book so you know exactly who lives in the city, but you completely lack the street map.
Which is pretty useless for actually seeing how the city functions.
Totally. You have no idea where they live, who they're talking to, or how they move around. And that is exactly the problem we are solving for you today on this deep dive.
It really is.
We are unpacking a breakthrough paper from the journal Science that finally, gives us that map. We're exploring a revolutionary new technology called Spatial VDJ.
Yeah, and the ultimate value of the knowledge we are going to unpack today is, well, it's context.
Context, right.
Because for a long time, immunologists have had that phone book of the immune system. We've been able to easily identify B cells and T cells, which are, the absolute heavy hitters of our immune defense.
A major player.
Exactly. And the power of these specific cells lies in their targeted reactivity. I mean, they don't just attack anything blindly. They're highly specific.
Right. They need to know exactly what they're looking for.
Right. And that specificity is determined by what we call clonally heritable antigen receptors.
Okay. Clonally heritable antigen receptors. That's a mouthful.
It is, yeah.
Yeah.
But you could just think of them as like unique molecular ID cards. They tell the cell exactly what disease or tumor to hunt down.
Got it.
But until now, seeing exactly where these specific cells were operating inside the physical geography of human tissue was just a massive blind spot.
And to really understand why this new spatial VDJ technology is such a huge deal, we first need to look at the frustrating limitations of the previous tools scientists were stuck with.
Yeah, the old tools had some major trade-offs.
Right. It's what I like to call the shredded ID card problem.
I actually love that analogy. It's a great way to visualize it. Let's look at the standard techniques scientists have been relying on up to this point. On one hand, you have single-cell RNA sequencing. Now, this technology is fantastic at reading those precise ID cards of the immune cells.
It reads the name on the card perfectly.
Exactly. Specifically, it reads something called the CDR3 region. That stands for complementarity determining region 3.
Right.
It's the hypervariable part of the receptor that actually makes physical contact with the target antigen. Kind of like the specific grooves on a key that fit perfectly into a lock.
Okay, so single-cell sequencing reads that CDR3 key perfectly.
It does, but to do it, you have to completely dissolve the tissue.
Oh, right.
Yeah, you essentially blend the city into a molecular soup. You get millions of perfectly read ID cards, but you lose all the spatial resolution.
Because you blended the city, so the map is just gone.
Exactly. The map is completely gone.
Okay, so to get the map back, I know scientists turned to spatial technologies, right? They started using spatial transcriptomics, specifically something called the Visium protocol.
Yeah, the Visium protocol, which is honestly a brilliant piece of engineering. You take a paper-thin slice of frozen tissue and you place it on a glass slide.
Sounds standard enough.
But this slide is covered in thousands of microscopic spots, and each spot contains specific chemical barcodes.
Okay, barcodes.
Yeah, so when the RNA from the tissue binds to the slide, it gets tagged with that spot's unique spatial barcode. It literally attaches a molecular zip code to the biological material.
So we have our map back. We know exactly what zip code the RNA came from.
We do, but here is the biological catch, and it's a big one.
Oh boy, okay.
The Visium technology captures RNA by grabbing onto its tail end.
Okay.
But that crucial CDR3 sequence, the name on the ID card, the grooves on the key, is sitting all the way at the opposite end of that microscopic RNA strand.
Oh, no.
Yeah. And during the standard laboratory preparation to actually read these transcripts, the protocol physically fragments the RNA. It chops it up.
Wait, really? It just chops up the very thing we need?
Yeah, the fragmentation severs the connection entirely. So you're left with the tail end that has the spatial zip code and the head end that has the CDR3 sequence, but they are no longer physically attached to each other.
Okay, let's unpack this for a second. So it's like we have a map of the houses and we have mail delivered to them, but the specific name on the envelope gets put through a paper shredder before we can read it.
That is exactly what happens.
But why couldn't we just use a different map? Like, if chopping up the RNA ruins the very thing we are looking for, why do scientists do it? Why not just skip the shredding step?
Well, this raises an important question. And the answer is basically because of the sheer volume of background noise.
Background noise from what?
From the cell itself. A cell is a chaotic, noisy factory. It's constantly pumping out thousands of different RNA transcripts just for basic everyday survival.
Oh, I see.
So these highly specific immune receptor transcripts we care about, they're incredibly rare, and they are fragile. If you don't fragment the entire library of RNA into smaller, readable chunks, the sequencing machines get completely overwhelmed by the background noise of the tissue. They just read the biological junk and miss the rare immune keys.
Wow. Okay, so the challenge was finding a way to fish out these specific fragile ID cards before they get destroyed without losing the zip code attached to them.
Precisely. You have to save the ID card and the zip code at the same time.
So just to recap our mental model here for you guys listening, we want to map immune cells in tissue. Single-cell tech dissolves the map to read the ID.
Right.
Visium spatial tech keeps the map but is forced to shred the ID to read it, so we're stuck. Enter Spatial VDJ. The researchers basically had to invent a clever chemical hook to bypass the shredder.
Yeah, they developed a really elegant two-pronged solution. They introduced hybridization capture probes.
Hybridization capture probes? Okay, what are those?
Think of these probes as highly specific molecular magnets.
Molecular magnets. Okay, I like that.
They are designed to seek out and bind only to the constant regions of B-cell and T-cell receptors. So before the aggressive fragmentation step can destroy the link between the ID card and the zip code, these magnets pull the exact transcripts we care about out of the complex molecular soup.
So it basically rescues them from the shredder.
Exactly. It enriches the sample, it preserves the spatial barcode, it preserves the molecular counters that let us tally up the cells, and crucially, it preserves the full-length receptor sequence.
That is brilliant. And what's interesting is they didn't just make one version of this tool, they made two.
Right, they did.
They created a long-read spatial VDJ, which captures the full-length transcripts for both B cells and T cells. And then they created a short-read spatial VDJ, which basically copies the DNA in a targeted way, but just for T cells.
Yeah, that's correct.
But wait, why bother inventing two completely different versions? Isn't having a short-read and a long-read version just adding, like, unnecessary complexity for the researchers using it?
It might seem like it, but it's actually about balancing accessibility with biological necessity.
Okay, how so?
Well, short-read sequencing is a standard workhorse in almost every modern lab. It's widely available, it's cheaper, and it's faster. So for mapping T cells, the short-read version makes this technology highly accessible to basically anyone.
Okay. That makes sense.
But the long-read version isn't just a luxury for B cells, it's absolutely mandatory.
Why only B cells? Do they, like, have longer RNA or something?
No, it's not about length. It's about how B cells evolve. When a B cell encounters a pathogen, it undergoes a process called somatic hypermutation.
Somatic hypermutation.
Right. It intentionally mutates its own genetic code, specifically the receptor sequence, to try and bind the target even tighter. It's rapid microevolution.
Oh, wow. So it's actively upgrading its weapon.
Exactly.
Yeah.
And to track that evolution, to see a B cell lineage changing and improving its key over time, you need to read the entire full-length sequence to catch every single tiny mutation. A short read would just miss those crucial changes.
Right, you'd miss the subtle upgrades.
Exactly. But what's great is that when the researchers compared the T-cell data from both the short read and long read methods, the results mirrored each other perfectly.
Which proves it works.
Yeah, it proved the fundamental accuracy of the entire system.
Okay, so the tool is built. We have our molecular magnets. But now they needed a test drive. They had to prove this map actually works in real biology.
Right, you have to test it in the real world.
So the researchers chose human tonsil tissue, which honestly makes a lot of sense to me because tonsils are basically classic training camps for immune cells.
Yeah.
These highly organized, distinct neighborhoods, right?
Yeah, the anatomy of a tonsil is the perfect proving ground for spatial technologies. The structures are very well known, and the scale of the data they retrieved was staggering.
Really? How much data?
This single experiment yielded over 62,000 unique B-cell and T-cell clonal sequences.
Whoa! 62,000.
Yeah, that is a massive density of spatial immune data.
But wait, earlier we talked about those microscopic spots on the slide having zip codes. Those spots aren't capturing just one single cell, right? They're capturing microscopic clumps of maybe 5 or 10 cells.
Right, it's not true single-cell resolution.
So if I have a spot with 10 different cells in it, how do they know if that specific spot is a B-cell neighborhood or a T-cell neighborhood? Like, it's just a mixed smoothie of genetic material.
It is a molecular smoothie, and this is where computational biology steps in to save the day. They used a mathematical process called deconvolution.
Deconvolution.
Yeah. Imagine tasting a complex smoothie and mathematically deducing exactly how many strawberries, blueberries, and bananas went into it just based on the flavor profile.
Okay, that sounds like magic.
It's just very complex math.
Yeah.
The software looks at the massive mix of RNA in that microscopic spot and calculates the probable proportion of different cell types.
Oh, I see.
And when they mapped it out, the spatial distribution matched the known anatomy of a tonsil perfectly. The B-cell clones were heavily concentrated in the B-cell follicles, while the T-cell clones packed right into the T-cell zone.
Okay, and they didn't just trust this brand new tool blindly, right? I read they ran a totally independent traditional sequencing method alongside it just to double-check their homework.
Yes, exactly. They used an orthogonal method called SMART-C3.
Right. And finding the exact same clonal sequences with a completely different method proved they weren't just picking up, like, chemical noise. The map was real.
It was very real. And beyond just finding the cells, they looked at how these clones were spread out.
Oh, this is the part I found fascinating.
Yeah, they found that expanded clones, meaning immune cells that had successfully recognized a threat and multiplied heavily, were detected spanning across multiple sequential slices of the tonsil tissue.
It is so cool. It's like looking at a wildly popular local restaurant franchise that has expanded to take up an entire city block.
That's a great way to think about it.
Whereas the naive, unexpanded clones, the ones that haven't found their specific target yet, they were just tiny pop-up stands found on only one street corner on one single slice.
Exactly. And if we connect this to the bigger picture, that spatial distribution tells a powerful biological story. Secondary lymphoid organs, like tonsils, are mostly composed of naive clones just waiting for their specific antigen.
Right.
So finding and mapping these massive expanded clonal families stretching across the 3D space of the tissue proves we can track an active, successful immune response in physical space.
Incredible. Okay, let's do another quick mental checkpoint for the listener. We've learned that Spatial VDJ rescues the shredded ID cards of immune cells using molecular magnets. We tested it in the tonsil, mapped over 60,000 clones, and proved it can track massive clonal expansions across a 3D space.
Perfect summary.
So the tool works beautifully in a highly organized, healthy tissue, but the real test for any immune mapping tool isn't peace, it's war.
Exactly.
What happens when we take this technology and drop it into the chaotic, disorganized battleground of a disease?
That is the ultimate question. Because moving from a healthy tonsil to a tumor microenvironment is a massive leap in complexity. Tumors are disorganized, they're chaotic, and they actively suppress the immune system.
Right, they fight back.
They do. So the researchers deployed Spatial VDJ on human breast cancer tissue to see if they could map the immune response actively fighting the tumor.
And they pulled it off. I mean, they identified distinct B-cell lineages that were spatially segregating along completely different areas of the exact same breast tumor. It's like different platoons of the immune system taking up different tactical positions around the enemy. But there was a major biological and mathematical hurdle they had to clear first to get that data, right?
Yes, a huge one. It's what we call the pairing problem.
The pairing problem.
Right. So an antibody, the actual weapon produced by the B cell, is made of two distinct protein chains, a heavy chain and a light chain.
Okay, heavy and light.
For a B cell to be functional, it needs both, and they have to match perfectly. But remember those microscopic zip code spots capturing clumps of cells?
Right, our molecular smoothie spots.
Exactly.
Right.
You might pull down 5 different heavy chains and 5 different light chains from one single spot. Because you lack true isolation-level single-cell resolution, figuring out which heavy chain goes with which light chain in that microscopic soup is mathematically daunting.
Yeah, I mean, so how can an algorithm possibly know which two chains go together? If they are all mixed up, isn't it just a guessing game?
It would be, absolutely, if we only looked at one spot. But the researchers developed a computational framework called REPAIR.
Repair, okay.
This algorithm solves an optimization problem by looking at spatial co-occurrence across the entire tissue section, not just one isolated spot.
Okay, let me try an analogy here to see if I get it. It's like a matchmaking detective at a massive networking event.
Okay, I like where this is going.
If you go to a party and you see 50 people in a room, you don't know who is married to whom, but if a specific heavy chain and a specific light chain continuously show up in the exact same spatial zip codes across the tissue, like if they show up to the exact same parties at the exact same times over and over again across thousands of spots, you can safely deduce they are a couple.
That is spot on. That is exactly the mechanism behind the REPAIR algorithm. By tracking the consistent spatial correlation of specific heavy and light chains across the thousands of spots on the slide, it pairs them up with incredibly high confidence.
That is so clever.
It really is. And this allowed them to reconstruct the full functional antibodies that were infiltrating the breast cancer tumor. It bridges the gap between just looking at spatial data and actually discovering functional therapeutics.
That is brilliant. Okay, so we've mapped the clones, we've paired the heavy and light chains in the middle of a tumor. But the ultimate power of this technology isn't just seeing where these immune cells sit, right? It's tracking how they change over time.
Yes, and this brings us to where spatial VDJ actually challenges textbook immunology.
Oh, really?
Yeah. As an immune response progresses, B cells undergo a process called class switch recombination, or CSR. Basically, they change their isotype.
Her isotype.
Right. They might swap their heavy chain constant region from an M type to a G type, for example.
Wait, what is the biological reason for doing that? If they already have the right key for the lock, why change anything?
Well, they keep the key exactly the same, but they change the handle. Swapping the constant region changes the function of the antibody entirely.
Oh, okay.
It might make it better at penetrating deep tissue or better at recruiting different immune helper cells to the fight. You can think of it like a police officer swapping a standard patrol uniform for heavily armored riot gear.
Okay, that makes total sense.
They're still hunting the exact same suspect. The target hasn't changed, but their operational capabilities have completely shifted.
Now, the standard biological assumption, basically what is written in the textbook, is that this uniform swap, this class switching, mostly happens deep inside the germinal centers, the heavily guarded B-cell follicles.
Right.
It happens inside the official locker room, so to speak.
That has absolutely been the prevailing model for a long time. But with spatial VDJ, because they have the long-read sequences and the spatial zip codes, they could look for these events directly in the tissue.
So what did they find?
Well, they scanned the data for instances where a clone had the exact same targeting key, but different functional uniforms, co-occurring in the exact same spot.
Okay.
And they found 17 of these putative class switching events. But fascinatingly, all 17 events occurred extrafollicularly, meaning outside the follicles.
Wait, outside. So they caught the immune cells changing their uniforms in the alleyway rather than inside the official locker room?
Exactly.
But why would they do that?
It points to a highly dynamic, rapid response mechanism. Perhaps in a fast-moving infection or a hostile tumor environment, the cells bypass the normal, slow vetting process of the germinal center to deploy a new weapon immediately.
That is incredible.
And to prove this wasn't just a technical glitch, the researchers actually looked at the physical tissue and found that the specific proteins responsible for rewriting the cell's DNA were physically located right there in those extrafollicular alleyways alongside the switching cells.
That is wild. And because they have the long-read sequences showing all those tiny micromutations, they didn't just catch them changing clothes, they actually tracked them moving across the city.
Yes. They built a spatial phylogenetic lineage tree.
A spatial phylogenetic lineage tree.
Yeah, they literally caught a specific B-cell subclone that had originated in one germinal center, let's call it follicle 19, evolved, mutated, and then physically migrated out to seed an entirely new germinal center, follicle 26, in a different part of the tonsil.
This is basically a time-lapse video of immune evolution, but extracted from a single frozen snapshot of tissue.
Exactly.
By looking at the mutations and where they are physically located, you can rewind the tape and see exactly how that clone expanded, mutated, and marched across the tissue.
It adds incredible nuance to our understanding of immunity. What's fascinating here is that tracking a cell's migration from one follicle to another proves this technology can reconstruct spatial lineage trajectories.
Right.
We are no longer just looking at a static list of cells. We are watching a dynamic spatial war unfold in front of us.
So bringing this all together, today we've seen how spatial VDJ completely changes the game. It rescues the shredded ID cards of immune cells using molecular magnets, linking a cell's molecular identity with its physical location on the street map of human tissue.
It really is a massive leap forward.
Whether it's the healthy training grounds of a tonsil or the chaotic battleground of a breast cancer tumor, this tool lets us see who is fighting, where they are fighting, and how they are evolving over time. But, why should you, the listener, care about this map?
Because knowing the geography of the immune response is the absolute key to the next generation of therapeutics.
Exactly.
Whether scientists are trying to understand why a SARS-CoV-2 infection overwhelms one patient's lungs but not another's, or trying to harness targeted T cells to attack a solid tumor, spatial context is everything.
It's about finding the specific immune cell that is successfully winning the fight in a specific, highly suppressed neighborhood of a tumor, figuring out its exact ID card, and then mass producing it as a customized therapy.
It really leaves us with a very provocative thought for the future, if you think about it.
What's that?
If we can now read the exact spatial blueprint of a successful immune response, literally mapping every clone and its evolutionary path across the tissue, how far are we from using this technology to artificially 3D print synthetic lymph nodes or custom tumor-fighting microenvironments perfectly tailored to a patient's exact biology?
Wow. From needing a street map to eventually building the city itself, that is definitely something for you to mull over. Remember, the world is way more complex and way more fascinating than it seems on the surface. Keep questioning the map, keep looking closer, and we'll see you on the next deep dive.